In this video, we'll show you how, during this experiment, we'll use the chromatography process in different types of leaves in order to see the different pigments there are.
The problem is, how can we separate the different pigments from the leaves?
Our hypothesis is that if we use the chromatography process on the different leaves, then we will get the different pigments because the chromatography process is used to separate a compound into individual components.
The materials we are going to use are chromatography solvent, chromatography paper, ethyl alcohol, spinach leaves, test tubes, fresh plant leaves, mortar and pestle, steering boa, paper clip, and a cork.
Control group, spinach leaves. Experimental group, plant leaves. Independent variable, different leaves. Dependent variable, different pigments observed. Constants, same solvent, same environment, and same paper.
Procedure. Leaves should be grouped by kind and color. Work with spinach leaf and with one or more other types. Number 1. Cut a strip of chromatography paper so that it just can fit inside a 15 cm test tube. Cut a point at one end of the paper. Draw a faint pencil line. Bend the paper clip and attach it to the cork stopper. Attach the paper strip so that it hangs inside the tube. The sides of the strip should not touch the glass. Number 2. Tear spinach leaf into small pieces. Put all of the pieces into a mortar. You can add a pinch or two of sand. This will help you with the grinding. Add about 5 milliliters of ethyl alcohol to the leaf pieces. Crush the leaves with a pestle until the mixture is finely ground. The liquid in which the leaf pigments are now for paper chromatography dissolved is called the pigment extract. With a glass rod, you will touch the substance, so you can leave a drop of the pigment extract to the center of the pencil line on the paper strip. You will let it dry and repeat as many as 20 times to build up a pigment spot.
The results we got from our experiment were: spinach has two pigments, xanthophyll and chlorophyll B, with RF values of 0.28 and 0.21. For the tree leaf, we had also two pigments, xanthophyll and chlorophyll A, with RF values of 0.21 and 0.39. And last, for the flower leaf, we had three pigments, anthocyanin, xanthophyll and chlorophyll B, with RF values of 0.4, 0.31, and 0.42.
Our conclusion is that, as we said in our hypothesis, The chromatography process that we did on the laboratory during our class shows the amount of pigments of each of the 3 plant leaves that we had for our experiment. We conclude that the plant with more pigment was the flower pink leaf. We discovered that we have made something wrong, because the measurements on the chromatography paper weren't as they were supposed to be. We supposed that it was because of time. We could upgrade our experiment getting more specific information, like for example on time or height we were supposed to base.